Welcome to the Neuroanatomy Series run by NANSIG. My name is Jiggy. I'm an F1 in the Oxford Deanery, and in this episode we'll be discussing cerebrospinal fluid and the ventricular system. Let's take a closer look at what we're going to cover in this video. So the brain sits around being bathed in cerebrospinal fluid, which is the special fluid that has lots of jobs, but we'll come on to those in more detail later. The brain needs lots of glucose and lots of oxygen to work, but it isn't in direct contact with the blood. It has the blood-brain barrier. And the blood-brain barrier is essentially a semi-permeable barrier that stops solutes in the blood from non-selectively crossing into the extracellular fluid of the nervous system. The collective term blood-brain barrier is used to describe four main interfaces between the central nervous system and the periphery. These four are shown in this picture. For the purposes of this video, we will refer to the blood-brain barrier proper. If you wish to learn more about the meninges, more about the cerebral vasculature, and more about the cerebral cortex itself, we have videos on those in this series, so definitely check those out. Let's take a closer look at the blood-brain barrier. It is formed by endothelial cells of the capillary wall, astrocyte end feet ensheathing the capillary, and pericytes embedded in the capillary basement membrane. Now, if you remember, astrocytes are star-shaped glial cells that perform loads of functions, such as structural support and metabolic support. Pericytes wrap around the endothelial cells that line capillaries and venules and help to maintain homeostasis in the brain, as well as sustaining the blood-brain barrier itself. They have a variety of other functions, like phagocytosis of cellular debris and maturation of endothelial cells, but you can read more about this in your own time. So we've established that the blood-brain barrier's role is to regulate what goes in and out of the extracellular fluid that surrounds neurons in the brain. Since it has these tight junctions between endothelial cells of the capillaries in the brain, it doesn't let large molecules, hydrophilic molecules, pathogens, or the passive diffusion of blood solutes into the CSF. The whole aim is to allow the diffusion of hydrophobic molecules like oxygen, carbon dioxide, hormones, etc., allow those through whilst actively transporting metabolic products like glucose across the barrier. The barrier also restricts immune factors like signaling molecules, antibodies, and immune cells into the CNS, thus protecting the brain from damage due to peripheral immune events like an infection. CSF is a clear fluid that circulates around the brain tissue and helps to maintain the brain's internal environment. We're going to go into more detail about the flow of CSF around the brain and what its role is. The first role of CSF that I'd like to talk about is buoyancy. The human brain itself is quite heavy. It weighs around 1.4 to 1.5 kilograms. However, because it's suspended in CSF, it actually equates to around 25 to 50 grams. Buoyancy is a key role for CSF because if the brain wasn't floating in CSF, it would crush all the important nerves and vessels around it. The next key role is protection. So CSF protects the brain tissue from injury. When the head is kind of jolted or it's hit, the fluid actually provides a buffer, kind of acting like a shock absorber from some forms of mechanical injury. And if you shake your head around while you're watching this video, you can tell that your brain is not slapping the sides of your skull. Let's go on to prevention of brain ischemia. It's aided by decreasing the amount of CSF in the limited space that there is inside the skull, and that decreases the total intracranial pressure and facilitates blood perfusion. Next, we have homeostasis. CSF allows for the regulation of the distribution of substances between the cells of the brain and neuroendocrine factors, to which slight changes can cause problems or damage to the nervous system. I like to use the example of glycine, where a high concentration of glycine can actually disrupt temperature and blood pressure control, and high CSF pH can actually cause dizziness and syncope. So it's really important that everything is kept in homeostasis to allow the CNS to function properly. Finally, the last key function of CSF is the idea of clearing waste. CSF allows for the removal of waste products from the brain, and it's critical in the brain's lymphatic system, which is actually called the glymphatic system. Metabolic waste products diffuse rapidly into the CSF and they're removed into the bloodstream as the CSF is absorbed. And when that goes wrong, the CSF can actually become toxic. And that's the case in such conditions such as amyotrophic lateral sclerosis, which is the most common form of motor neuron disease. So if the CSF just sat around the brain, it wouldn't be able to get to all the important parts inside of it. Therefore, the CSF also flows throughout the brain in these hollow structures that we talked about before, called the ventricles. These are the lateral ventricles, of which there are two, the third ventricle and the fourth ventricle. And these ventricles actually make CSF in areas of cells called the choroid plexus. But let's delve into how CSF is made. CSF is made in the choroid plexus. The choroid plexus consists of a layer of cuboidal epithelial cells surrounding a core of capillaries and loose connective tissue. The epithelium of the choroid plexus is actually continuous with the ependymal cell layer, which is also known as the ventricular layer, that basically lines the ventricular system. The choroid plexus folds into many villi along each capillary, creating frond-like processes that project into the ventricles. The villi, along with a brush border of microvilli, increase the surface area of the choroid plexus. CSF is formed as plasma that's filtered from the blood through the epithelial cells. The choroid plexus epithelial cells actively transport sodium ions into the ventricles, and water follows due to the resulting osmotic gradient. Let's now talk a bit about the flow of CSF through the ventricles and around the brain. What becomes dilated and the symptoms that come from it often depend on what is being blocked. For example, if the foramen of Monro is obstructed, then that might lead to dilatation of one, or if it's large enough, both lateral ventricles. The foramen of Monro, by the way, which you'll sometimes hear people say, is just a fancy name for the interventricular foramen that exists between the lateral ventricle and the third ventricle. If the cerebral aqueduct itself, which is normally narrow anyway, becomes obstructed, then that can lead to dilatation of both lateral ventricles as well as the third ventricle. I like to think of the whole system as kind of like a pipeline, and if somewhere in that becomes blocked, then the places that are behind that blockage will become dilated. Depending on the cause, a shunt can often be put in to drain the extra CSF into the abdominal cavity. This is called a ventricular peritoneal shunt. With this is just to divert the extra CSF and reduce the pressure inside the skull. The last clinically relevant area I'd like to talk about is CSF analysis, and we do this via doing a lumbar puncture. A lumbar puncture can be used to analyse the CSF, most commonly for things like infectious diseases of the brain and spinal cord, including meningitis and encephalitis. It is also helpful in autoimmune disorders such as Guillain-Barré syndrome and multiple sclerosis. You may hear it be called a spinal tap, although that's a bit too American for this video. The ideal insertion point of the needle should be either in the interspinous area between L4 and L5 or L3 and L4. The CSF can then be withdrawn and analysed. Under normal conditions, the composition of the CSF should remain constant, but in various neurological diseases, especially in acute conditions, the composition, quantity, and its pressure can be altered. By measuring the levels of various CSF components as shown in this table, the diagnosis, severity, and prognostication of neurological conditions like infections, subarachnoid hemorrhage, demyelinating conditions, tumor-like conditions, etc., can all be done. In terms of exam questions, it's definitely worth remembering this table. And a key point to also remember that you can see at the bottom is if you see the question involves blood or xanthochromia in the sample, then I would be thinking about subarachnoid hemorrhage. So that's it. Thank you so much for listening. I hope you enjoyed this introduction to CSF and the ventricles. Just a few further bits about, reading if you want to go and learn more about this topic further. I really enjoyed looking at an MRI cyst flow on YouTube, and I've put the link there, but you can also just type it into YouTube. Essentially, it's a way of imaging that actually shows you the flow of CSF through the ventricles. For some reason, I couldn't pop it into this video. It wasn't working, but I would recommend it. You do go and check it out. Geeky Medics, as always, is what I used for the CSF interpretation slide, and they always have really good content. Teach Me Anatomy is really good for the basics of neuroanatomy. If you want to go into a bit more further detail, or you enjoy videos and descriptions, then Dr Najib lectures on YouTube are always a classic. Most medical students and foundation doctors can use those. They're always really comprehensive, and same with Sam Webster Anatomy. He does some really good explanations too. So If you feel like you do need to go and get some more information, then definitely check those out. I'd like to also mention that we're extremely thankful for the support of Mr Kaliaperumal, who's consultant neurosurgeon at Edinburgh Royal Infirmary in the Department of Clinical Neurosciences. He checked over this video and gave some tips and tricks and signed off on it. So thank you so much to him. And lastly, thank you to you, everyone who's watched this video, who's watched all of our other videos. We really did want to kind of put together a really comprehensive neuroanatomy course for you all. So we hope that you've thoroughly enjoyed it and, feel inspired to go on and learn more and more about neuroanatomy. We all find it really interesting, and we really hope that you have enjoyed it.